All right, welcome everyone. Today we're going to be talking about whole genome compared to genomics, and specifically genome evolution. So here we are in the second lecture of module 5. On Tuesday we talked about comparative genomics and using evolution to understand genomes. Today we're flipping this around and we're using genomes to understand evolution. So this is the second part of comparative genomics, and we're going to be talking about big events that happen. And then next week we're going to be talking about phylogenetics and phylogenomics. So as I mentioned on Tuesday, we've entered the age of comparative genomics. There are hundreds of mammals now, hundreds of yeast, hundreds of flies that are sort of constantly being sequenced across many different centers, enabling us to ask questions that were never before possible. So what we're going to be looking at today is what are some of the computational underpinnings of these questions, and what are the techniques that we can use to understand these genomes, and specifically to study genome evolution, both near and far. So the first thing that you need to do when you sequence all these additional species is to assemble the genomes. And that's not a trivial problem. So first we're going to talk briefly about the first foundation to comparative genomics, which is building a good assembly. Because if your assembly is not good, then anything that you infer as evolutionary event might actually just be simple assembly errors. The second foundation to whole genome comparative genomics is really whole genome alignment. So being able to actually generate alignments of the resulting sequences on top of each other to basically understand what nucleotides and what regions correspond to each other across species in order to understand the events that have separated them. After we've covered these two foundations, then we're going to talk about super distant evolution and what could be thought of as an evolutionary jump, namely the process of whole genome duplication. And this is not just some esoteric process that doesn't concern us. Every one of you guys, and I'm looking at you, underwent 2 rounds of whole genome duplication well before you were a vertebrate. So your great, et cetera, ancestor underwent 2 rounds of whole genome duplication that gave rise to the complexity of body plan that we see in vertebrates today. And we talked briefly about the Hox clusters last time. This is the first round of evidence for the 2R hypothesis. But then the more recent evidence came from the techniques that I'm going to describe in part 3. Then we're going to shift gears and talk about recent evolution, so human relatedness. We're going to look at how we can measure selection within the human population at different scales. Selection very recently, more distantly, very distantly, and so on and so forth. Then we're going to talk a little bit about human demographic history, namely, who are we and how did we get here kind of questions. Looking at some work by David Reich more recently. This is by Pardis Sabeti and others. And then we're going to be looking at human relatedness and ancestry painting and tying all this back up to GWAS and these normalization factors that we talked about, normalization techniques that we talked about in EQTLs. Who's with me so far? Awesome. All right, so first part, building the genome assembly. And these are slides that I have inherited from Serafim Batsoglu. So this is kind of cool. At the time when we were assembling the human genome, there are obviously 3 billion nucleotides. I don't know how many of you guys have put together a puzzle with 3 billion pieces, but finding a corner is not obvious. So how do you even assemble the genome? So the way to sequence genomes, unfortunately, involves cutting them. It's not that we like making puzzles, so we take these beautiful pictures, we chop them up, and then we reassemble them. No, we have to cut the genome. And the reason for that is that our sequencing technologies simply stop working well after, in the old days, 1,000 nucleotides. In the more recent, newer, and improved technologies, about 70 nucleotides. No, I did not get that wrong. It is actually that way. So the old sequencing technologies used to get about 1,000 bases. The new sequencing technologies get about 70 bases. And the problem is, the big difference, of course, is the cost. I'd rather get 70 bases at a cost of a genome per 1,000 nucleotides, per $1,000, rather than get 1,000 bases at the cost of a genome per $1 billion. Okay, so it's maybe a factor of 10 smaller, shorter, but it's a factor of a billion cheaper, or a million cheaper. The way to carry out genome sequencing involves cutting the genome in order to be able to get sequences across these short fragments. And the reason for that is that in the new next generation imaging technologies, things stop working as well after a few dozen nucleotides being incorporated. And in the old sequencing technology, you simply can't tell the difference in weight between 1,000 nucleotides and 1,001 nucleotides. So what do? You first cut the genome many, many times at random in order to be able to sequence the ends of these fragments. Notice that everything is paired end sequencing, okay? Every single time you make one of these fragments, it has two sides, both of which have a little overhang. And in both cases, you're sequencing from the 5' to the 3' direction in this side and that side of it. Everybody with me here? So you basically obtain either 100 base pairs or 1,000 base pairs of the end of a fragment, and you know that it is paired to another fragment that comes from the same location in the genome, pointing in the opposite direction, okay? Moreover, if you construct your library, your genome library right, you end up with fragments that you have selected to be a certain length. So if I select fragments that are about 40,000 nucleotides long, or 2,000 nucleotides long, or 1,000 nucleotides long, then I can say something about the distance at which these locations should be mapping to the genome. Raise your hands if you're with me on that concept. Perfect. So basically, I first make fragments, then as I select them for different library sizes, and then I sequence forward and reverse and reads. One of them should be mapping on the Watson strand, the other one should be mapping on the Crick strand of my genome assembly. So one of them should be mapping as a reverse complement, and they should be pointing to each other. If that's not the case, and if they're not at the right distance apart, and if they're not pointing in the right direction, if they're like that, or like this, or in different chromosomes, you know something wrong happened with that segment of DNA. And therefore, you can simply ignore that fragment as being what we call chimeric. A chimera is an ancient Greek animal that had the head of a lion and the wings of an eagle and all kinds of other weird things that was basically made up by chopping together parts from different animals. This is why we call this a chimera, because it's basically taking parts and mixing them together. The origin of the word is probably chi, the letter chi, which basically involves a crossover, and then mer, which basically means parts. Okay, so it's like a mixing of different parts. You had a question?
Just for like terminology, so like what is Watson strand versus the Crick strand?
The Watson strand is the slightly taller one, and no, it's arbitrary. Basically, the Watson strand is usually the forward one, the Crick strand is the reverse one. It's just to honor the two guys who kind of stole the stuff from Rosalind Franklin. Sorry, I'm being rude, but no, they're awesome scientists, both of them. I won't talk about person. Anyway, other questions? Okay, so how do you now put these pieces back together? You now have all these little pieces. You kind of know how they relate to each other in a pairwise fashion, but you don't know the whole layout of the puzzle. So one way to do genome assembly is to first overlap, then lay out whatever you overlap, and then build a consensus out of that. So the idea is that you compare, kind of like when you're building a puzzle in your living room, you basically compare pieces to each other, and every time you find a match, you basically now have two pieces that are joined together. And then after you've continued building them, you basically have what we know as contigs, for contiguous blocks of sequence. And then you can use the spacing information between your paired end reads to figure out the layout of these contiguous chunks, okay? So these are individual reads. Based on the overlap, you build contigs, and then you can put the contigs in a relative order to each other and build super contigs or scaffolds, okay? And then at the end, you derive the consensus sequence from that. Raise your hands if you're with me. Awesome, great. So a read is about 500 to 900 bases long with the old technology, about 70 bases with the new technology. A mate pair is a pair of reads whose two ends come from the same original fragment. A contig is a contiguous sequence. A super contig or a scaffold is what you get after, and the consensus sequence is what you produce at the end. Okay. So how do you find overlapping reads? You could basically sort all of the k-mers in your reads. You can find pairs of reads that share a k-mer, and then extend to the full alignment, and then throw away if they're not very similar, because it basically means that you have some other part of the genome that you're now assembling. How do you merge the reads? You basically have to infer where are the boundaries of contiguous segments, and sometimes you may have bifurcations. So here, I'm very happily building my puzzle, putting piece after piece after piece after piece, and then I realize that some of the pieces join to the blue sequence over there, and some of the pieces join to the red sequence over there. Up until here, they were identical, but after that, they're actually different. So what I thought were a single sequence, I'm now realizing is a double sequence, okay? And that's due to repetitive regions. This could just be some of the ancient repeats that make up about, I don't know, 30% plus of the human genome. This could just be a recently duplicated gene or a gene family that's there in tandem, and so on and so forth. So there's a lot of repetitive elements in the human genome, and every single time they're longer than the length of your read, you're kind of in trouble, because you have to infer how they eventually separate out. Raise your hands if you're with me so far. Awesome. So what are some indications that I have over-collapsed region? So this region is over-collapsed because collapsing reads that come from the same place is collapsing. Over-collapsing is basically when you take places from different parts of the genome and you put them on top of each other, okay? So this region is over-collapsed because, of course, at the end of it, you bifurcate. But also, if you look at the coverage, the average coverage of your genome, if you look across the whole genome, you might have, I don't know, you might have sequenced the genome at 50-fold coverage. So at every nucleotide, you have on average 50 reads piling up, and it follows some kind of Poisson distribution. But in some locations, you might realize that, wow, I have 100-fold coverage here. I'm doing great. Or I'm over-collapsing. Everybody with me? So you can kind of tell these regions because they have a lot more reads than what you would expect. Everybody with me? Awesome. So you now know that you're in an overcollapsed region. You now know that you want to resolve it, but you might not have the right information. So what information could you use to resolve it? Well, maybe you have some paired end reads from much longer fragments that basically say that red is actually going with red here, because I have a paired end read that uniquely maps here and uniquely maps there. If I only have something that's paired end, uniquely mapping here and uniquely mapping there, it's no help, right? Because here I cannot tell blue from red. Whereas here I can tell them apart, and here I can't tell them apart. So knowing whether blue goes to blue or blue goes to red is extremely important. Everybody with me so far? So one way to do that would be to have libraries of multiple sizes. So one approach is to actually build both a plasmid library with a 10 kb and a cosmid library with a 40 kb and a phosmid library with 100 kb, so that I have junctions at much longer intervals, which would allow me to resolve ambiguous repetitive regions. Everybody with me so far? Awesome. So we've now merged these regions into contigs, and we basically merge them up until potential repeat boundaries. And then we just stop the merge there, and we stop the merge here. And we basically say, here's a contig. Here's a contig that I know is over-collapsed, but I'll deal with it later. And here's another contig. Sounds good? So once I've done that, I end up with a graph. A graph that basically tells me that this contig here is linked to either this contig or that contig on this side. And then this contig here is linked to either this contig or that contig on that side. And what I need to solve now is a graph traversal problem. I need to go from one end of one chromosome all the way to the other end of the same chromosome, traversing these links and walking through that graph in a linear fashion, okay? And then I might realize that this overcollapsed contig, in fact, maps one time in chromosome 3 and one time in chromosome 17. In context of 3, it has the blue sides outside, and in the context of 17, it has the red sides outside. Sounds good? So I basically have my graph, and I need to resolve it, and I have to basically, you know, remove you know, these ambiguous things. And what I want to do is traverse my genome exactly once. So it's kind of like, you know, graph traversal problem where I have to walk through every path once. Okay. So basically, this allows me to now resolve the 2 copies of this particular repetitive region. Okay. So again, repeats that are shorter than the ring length are okay. Repeats that have more base pair differences than the sequencing error are okay. But to make the genome appear less repetitive, you can either increase the read length or decrease the sequencing error rate. And you know, you can do a lot of error correction. Basically, usually you discard a vast majority of sequencing errors that way. And then to build your consensus sequence, you have to either ignore polymorphisms or embrace polymorphisms. Basically say, well, I'm just trying to build a consensus. I'm just going to take the major allele. Or A, I actually care a lot about these. And when you're doing clinical genome sequencing or when you're mapping genetic variation for EQTLs across individuals, that's where you really care about these SNPs. Okay? So you want to derive the consensus based by some kind of weighted voting or take some maximum quality letter and so on and so forth. So that's one way to do genome assembly. Consensus, layout, overlap. The other one is to just go embrace the concept of the string graph completely and fully. Namely, you can basically build these De Bruijn graphs that basically contain every unique k-mer and the graph, how they're linked together, which is a big major shift in genome assembly that happened, you know, around the mid-2000s. Okay. So the concept of the string graph is that you want to be able to sort of argue about all of the possible ways of traversing it and resolve it. And there are many different types of tours that you can do. For this particular graph, where you know you have a unique contact here, unique contact here, and unique contact here, but all of these different ways of getting in and out of them, you basically have multiple solutions and they correspond to different versions of the genome, of course. So given a shotgun data set of reads, we should be able to build such a graph, and then we should be able to resolve this graph. So the string graph approach basically says, let's ignore these very long contexts. Let's simply chop up the contigs into little pieces and then build this graph of all of them, and then start reasoning about how to traverse this graph. And basically the way to traverse this graph is to have rules that resolve these connectivities based on arguing that the total number of reads going in and the total number of reads going out should be the same, and I should be able to sort of somehow add the same flow in and out of every edge, in and out of every contact. And therefore, if there's at least 1 going out and at least 0 going out, but the total here is 1, that allows me to somehow resolve the graph and basically say that this cannot be traversed. So there's a lot of arguing about graphs that allows you to sort of resolve these cases. And this is an art. You have to just figure out what are all the special cases and how do we deal with them and so on and so forth. Everybody with me? The other problem, of course, is that there are chimeras. There are chimeric reads that somehow have, in my sequencing reaction, rejoined when they shouldn't have. And these can really mess up an assembly, and you can somehow infer from this string graph analysis which ones they are and resolve them. Okay. So then at the end, you're just trying to achieve a feasible flow, and you can argue about forward edges, reverse edges, in edges, out edges, and resolve all that. Okay. You don't have to worry about all of the details, but this is just important to know. Everybody with me on part 1? Great. So now that we've assembled our genomes, let's start aligning our genomes. Sounds good? So how do we do whole genome alignment? We talked about dynamic programming as the basic foundational mechanism of alignment, where basically you're sort of somehow traversing a graph from one end to the other end. With multiple genomes, you could do the same traversal, but you're not looking at 2 dimensions anymore. You're now looking at a traversal in 3 dimensions, where you're adding a gap in this species, or in that species, or in the 3rd species in the z-axis, or a gap in 2 of them, and so on and so forth. Okay? So the same exact dynamic programming algorithm generalizes to 3 species, and to 4 species, and to, you know, k species. But the problem is that the corresponding algorithm is now n to the k instead of n squared, okay? And n to the k, if you're dealing with 1,000 base pairs and, I don't know, 100 species, is a very, very big number. So you want to avoid multiplying by 1,000 100 times. So the typical way of doing this is through a tree where you do progressive alignment, where you basically first take the two closest species, you build some kind of consensus, you align the next one on top of it, you align the next one on top of that, and you sort of build that in a nested fashion going up your tree, enabling you to, in the end, get a consensus for the whole thing and map them on top of these intermediate internal nodes. Raise your hands if you're with me. Awesome. So there are many algorithms for building these alignments, these global alignments, but as we mentioned, there may be rearrangements. In fact, they are very common across different species when you're doing comparative genomics. So what you would like to be able to do is find local alignments and then chain them together and carry out restricted dynamic programming between them. So the idea is that instead of doing a dynamic programming on the entire matrix, what I could do is find high scoring pairs, and then only build the dynamic programming surrounding these segments. So basically, I found my high scoring matches. I now have some way of traversing this sequence. And what I need to do next is deal with these lower similarity regions. Raise your hands if you're with me. Awesome. So here's what's interesting about these lower similarity regions. They're kind of crap to start with. You don't want to spend most of your algorithmic power trying to align things that don't really go together, okay? So if you have these alignments here, chances are your functional elements are going to be in the green parts anyway. So most of your algorithmic time appears to be in the blue parts right now because that's sort of where you're building the full DP matrix. Whereas here you're using these k times n bounded dynamic programming algorithm that we talked about in lecture 3. Everybody with me? So there are ways of overcoming this, and basically you should understand the trade-offs between accuracy, where the signal is, what you're trying to get out of the alignment, etc. If your goal for the alignment is to study highly conserved regions, who cares about the blue stuff? If your goal is to study evolution and you want to find very distantly related genes and exactly how they evolve from each other, then absolutely spend the time you need in the blue parts, okay? So every single time you develop some computational biology program, you should think, what is my end goal? What is the biological question that I'm trying to solve? Sounds good? All right, so as I mentioned earlier, progressive alignment is the way to go. Basically, given n sequences, instead of solving the length to the n problem, you basically want to do things progressively and basically go up the tree. So what I mentioned now was a way of going through the whole sequence in an algorithmically fast way that kind of ignores the full n squared matrix here and just builds a series of little matrices, and even those you might not care too much about, okay? But that's still assuming a single linear path from one end to the other end. This is still the kind of thing that you could get with your dynamic programming algorithm, the Smith-Waterman or the Needleman-Wunsch version. Instead, you might, however, have a rearrangement, and you might have a duplication, okay? So when you're trying to align this genome to that genome, there might not be such a simple path. So what you would like to do instead is build together these local alignments and nest them together. So a local aligner would basically be able to capture all of these, but would not have a common path going through them. And a global aligner would basically build some kind of really poor alignment here where there's an inversion, and a really poor alignment here where there's a duplication and no corresponding sequence, and try to still traverse it diagonally. Everybody sees the difference between these algorithms? So we'd like to have something that blends the two, that basically both gives us a global alignment, but also does well locally. Sounds good? So basically, combining global and local, you get basically glocal alignment, which is a portmanteau. But the goal is to find the least cost transformation of one sequence into another sequence using an additional set of operations, where instead of just having sequencing edits and inversions, you also have translocations, duplications, and combinations of the above. Everybody with me? So now you can extend your algorithm to have some kind of outer loop that does all of these things, and then is able to sort of chain together these regions. Sounds good? So this is basically the kinds of results that you get. If you only do local alignment, you have a lot of noise. If you do global alignment, you basically capture all of these inversions while at the same time painting a path through the whole sequence. Everybody with me so far? So we talked about resolving basically these region correspondence with global alignment. Let's now talk about how, when you're trying to study evolution, you can find the matching of genes to each other. So let's talk about gene-based global genome alignment. And again, the goal is to now use genomes to study evolution. So one way, so as we mentioned earlier, basically what you want to do is build an alignment of all the genes and all of their flanking regulatory regions. One way to do that is to basically try to find what is the matching gene for every region of the genome. Namely, if I have a gene here in one species, how do I find the corresponding gene in another species? So you basically want to look at amino acid similarity of the genes themselves. You want to find the locations of those genes. You want to know if they translocated, if they moved. And what makes it hard is that not all the regions have a one-to-one correspondence. There are gene duplications, gene losses, there's gene divergence, there's genome rearrangement. So what you'd like to do is find subgroups of regions that you know should be aligned together, even if they're not one-to-one. And that's sort of what this BUS algorithm does, this best unambiguous subgroups algorithm. It basically seeks to find the correspondence of both genes and regions using the complete bipartite graph connectivity between any pair of genomes and integrating both protein similarity and gene order information. And when you do that, you basically can infer orthologous relationships, you can infer duplications, you can infer losses, you can infer merges of genes by building a graph, a bipartite graph of your sequences, inferring from the similarity score matrix what are best alignments in every direction. And in this particular case, saying that, hey, it seems that these three genes here, A, B, C in one species and 1, 2, 3 in another species, are in fact arising from two different duplication events. A duplicating to become 1 and 2, and then 3 duplicating to become B and C. Everybody see that? Whereas if I was doing my alignment in a one-to-one fashion, I would end up with, I don't know, A and 1, C and 3, and maybe B and 2 would be left, and I would be having a very poor alignment between them. Everybody with me?
How do you turn the two sequences into a bipartite graph?
You basically just blast every gene in one species against every gene in the other species. And then you find connected components of that. Basically, that gives you the bipartite graph of who matches whom with some weight. And then you can find connected components in that graph by sort of removing all of the, basically by partitioning the graph, basically, some things will not match at all. And then you can start separating out in those best unambiguous subgroups by finding these locally maximal similarity scores. Everybody with me? Great question. Other questions? All right. So, and then once you've built this full bipartite graph, you can sort of find this directed graph that basically tells you who has the best match in which direction. And then 2 may have its best match in A, and B may have its best match in 3. And then you can sort of separate these components, and then you iterate. You just feed it through multiple cycles now that you've removed these edges. So when you apply this to these two species of yeast, what you end up with is initially so many matches. So basically, when you do blast all against all, you end up with pretty much, you know, very, very many off-diagonal hits. And as you start pruning and pruning and pruning, you end up with pretty much a straight line. That basically tells you that I've now successfully aligned all 6,000 genes in one species to all 6,000 genes in the other species. Everybody with me? So you know that we're comparing orthologous regions, and you basically can now generate a visualization of the gene order in these regions, and you can see how the genes are basically matching the right order in orientation. And every now and then you get some weirdnesses where some genes have duplicated and some regions have been lost and so on and so forth. Sounds good? So that's sort of the basis. And now you can use that basis to, yeah. So by basically building these extensions where you're basically asking what are all of my best matches in both directions, and then building connected components from the best matches. So you're basically going from A to all of its best matches, from 2 to all of its best matches, and you're preserving all of these best matches. And if I want to be friends with Jane, and Jane wants to be friends with Peter, and Peter wants to be friends with me, we end up in a clique altogether.
Yeah, I guess the plot, or the really noisy plot, is this if you superpose chemistry elastic on top of each other, and then you get a cleaner one.
That's exactly right. So basically the pruning happens through different stages of running this algorithm, where you're basically doing the, you know, the maximal out edges, and then some relative threshold that basically says, well, if Peter really likes me much more than he likes Mary, and so on and so forth, then, you know, we're going to sort of prune that edge because Mary already likes, I don't know, Sam much more, and so on and so forth. Does that make sense? Great. Where, of course, all these names are regions in the gene. Okay. Genes are much cuter when you give them names. As opposed to, I don't know, YIL060W. Although YIL060W is much more meaningful. Y for yeast, I for chromosome 10, L for the left part of the centromere, 060 for the 60th orth out, open reading frame out, and W for Watson. Okay, yeah? I'm sorry, Greek letters. It's probably something different in Roman letters. What chromosome should it be? There you go. Yeah, that sounds better. Sorry. So it's A, B, C, D. Okay, A, B, C, D, E, F, G, H, I, 9. Thank you. All right. Although I and J in Latin were the same letters, you can understand where the Greek is confused. The Romans invented I. I mean J. We don't have it. Anyway, everybody with me? And I actually used to be letter 10, the number 10 in Greek letters, because we actually had one more letter stigma as number 6. Sorry. All right. So all this to say I was wrong. Sorry. Thank you. All right. So then, having done this work, you can look for regions like this where things are in the right place, or you can look for exceptions. And what's really cool with looking at many closely related species, you can see evolution in action. You can basically see what changed in the evolutionary distance between human and mouse. And what changed can give you insights about the mechanisms of genome evolution. So for example, at the ends of the chromosome, you kind of are walking towards the end of the chromosome and you're finding this beautiful, beautiful alignment. And then you get near the telomere, and what happens? This. In these subtelomeric regions, you basically have these family expansions of genes that have duplicated all over the place, all kinds of insertions, duplications, exchanges between chromosomal arms, and so on and so forth. Turns out that the families, the gene families that are sitting in these regions, are actually involved in environment adaptation. So in the case of yeast, they are cell surface adhesion proteins, and you need to adhere to different sort of surfaces. If you're different species, if you're adapting to a new environment, you need to be able to evolve these families much, much more rapidly, expand them, you know, mix them up. And that turns out to be happening specifically in these regions of the genome. So it's kind of advantageous to have the genes that are associated with environmental adaptation in the places in the genome that are probably the easiest to break, mess up, duplicate, and so on and so forth. Everybody with me? So you can also look at evolutionary action. You can basically see, well, besides this telomeric expansion, what kind of other events are there? And you can actually recognize chromosomal exchanges where one chromosome is aligning here, and then suddenly, boom, another chromosome is aligning there. And you can basically ask, well, what happens at the boundaries? And it turns out that at the boundaries, you basically have these repetitive elements, these duplications that might actually serve as an evolutionary landmark upon which you can then carry out these translocation events, these chromosomal exchange events. Similarly, you can find inversions. You can basically see that these, you know, you're kind of aligning happily here, and then suddenly all of these genes are flipped in the opposite order. What's going on there? Turns out that every one of those boundaries has instead flanking tRNA genes in opposite transcriptional orientation. And you can imagine how the genome could actually fold onto itself and then flip that around. So you can actually find evolutionary events demarcated by these evolutionary operators, if you wish. You can also find these segmental duplications where several genes from one chromosome have now jumped into another chromosome. This is my favorite. This is an insertion of an intene. An intene is kind of like an intron, but at the protein level. So it's an intene. It's something that gets spliced out post-translationally. And this turns out to be a virus. This is a protein that gets inserted with a motif that favors it being inserted in the right reading frame so that it then translates itself. And when the host gene gets translated, that protein structure folds, catalyzes the capture of its own RNA and its reinsertion somewhere else in the genome. I mean, you know, if you can imagine it, nature has done it a few times. You can also ask, what is the difference in gene content between these closely related species? For example, we found that there were only 8 to 10 genes that were unique in each genome, and these were largely related with, again, environment adaptation, metabolism, regulation, stress adaptation. There were very few tandem duplications, 1 to 2 genes at a time, about 10 to 20 events, and then 2 larger segmental duplications, and also massive protein family expansions, which were primarily happening at the telomeres. Okay. You could also find places where there appears to be 2 different compensatory frame shifts leading to this super rapid evolution of this intervening sequence. So if you want to create completely new protein domains, well, why don't you duplicate a protein, keep its beginning and its end, and then evolve a new domain by sort of effectively starting with some scrambled sequence by translating a sequence in the wrong reading frame. You could also, we also found that there was a lot of stop codon loss and gain, leading to expansion of additional regions. And I mentioned these intene as well. All right, so basically what we're learning is these, you know, large evolutionary events that are happening at the distance of roughly human to mouse, about 60 million years divergence, okay? When you start going further in evolutionary time, you can basically see these much more dramatic events appearing every now and then. And one of them is whole genome duplication. So this is actually a funny story. We had basically ordered 4 yeast species from the strain repository to sequence them, and we had selected them to be roughly the distances of human, mouse, rat, dog, in order to basically build a model for what the mammalian comparison compared to genomics would look like. But one of the species was mislabeled, and it turned out to be much further in evolution. And we only figured out that it's mislabeled much later on, after we realized that there was something wrong with the karyotype from what we were expecting. The number of chromosomes was off. But that turned out to be a super lucky mix-up, because that species turns out to have preceded this whole genome duplication event, okay? And remember I showed you this beautiful picture of sort of, you know, starting with a very messy plot and then resolving it down to a straight line? Well, this species here did not resolve to a straight line. It was messy. And I used to think it was this nice, close related relative, but it turned out to be much more distant related. And I told my collaborators, Bruce Buren and Eric Lander at the time, that I would figure it out within a month. Let's write the other paper, submit the other paper, and then I'll come back to that. And I was about to leave for vacation, and it was like December 28, and I was flying out the next morning, and I was like, I have to figure this out. So here I was in my MATLAB script, trying to figure out what goes on here and why is this so messy. And I was zooming in different directions, and the thing that hit me is when I zoomed in this direction, where I just expanded it out, okay? So we have the chromosomes and all of the genes, all of the chromosomes end to end, all the scaffolds end to end for these species, K. walti, and all of the chromosomes end to end for Saccharomyces cerevisiae. Notice here I don't have chromosome numbers because we frankly didn't know what the chromosome boundaries were. The genome assembly was still a little messy. But in this particular case, we have the 16 chromosomes of yeast, of Baker's yeast. So now the question is, was my assembly messed up? Was my alignment messed up? Or was my, model of what separates the species messed up? And when you zoom in this way, what do you notice? What you notice is that this region here of Saccharomyces walltii appears to be mapping to two different chromosomes of Saccharomyces cerevisiae, to chromosome 7 and to chromosome 10. That was weird. And that was not just true for one region. Everywhere I looked across the chromosome, and it's now 11 p.m. and I'm alone in the lab at night, and I'm seeing this, and I'm like, I hope I don't die, because I want to tell people. So basically, I'm sending this email that basically starts describing this, point after point after point, and painting this picture of what could have happened here. And it gets weirder than this. Okay. If you look at the little dots, these are the genes. And those genes were, in fact, again, mapping in two different chromosomes. So here's my new species, and here's where it maps. And that's the first time that I realized, wow, these boundaries happen at the gene boundaries. There's nothing wrong with my assembly. Because I just told you how messy assembly can be. This could have just been a misassembly, just a bunch of, errors happen. But no, the transitions were in fact between gene boundaries. So I've already told you the answer, but why don't you raise your hands and tell me what could have happened here? Who can see what could have happened here? Raise your hands. Who cannot see? Raise your hands. Who has no arms? Raise your hands. Come on, everybody with an arm, raise your hand. All right, you have to raise your hand either when I say A or B. A, who can see what's going on? Okay, I have 3, 4, 5 answers. Who cannot see what's going on? Please make me happy. Awesome. Having people who cannot yet see what's going on is kind of exciting, because that's who I was. I was like, what the heck is going on here? So someone who can see it, tell me what's going on. Yes, that was the title of the section. Yes, that's exactly right. So it turns out that, and this is a cool animation. Are you guys ready? I'm going to do it twice. Okay. Okay. So if I now put Saccharomyces walltii in the middle, you can actually kind of start seeing what happened a little better. All of these genes were initially duplicated. And after that initial duplication, I then lost this gene, that gene, that gene, that gene, that gene in one copy, and this gene, that gene, that gene, that gene, in the other copy. Okay? Which genes did I lose completely? Raise your hand if you can see them. Which one did I lose completely? Which one? Aha, none. It's a trick question. All the genes are there. It's amazing. Why are all the genes there? Raise your hands if you can see why are all the genes there. You have one answer, 2 answers, 3 answers, go for it. Yes, but why are all the genes preserved? Yeah? They're kind of important. I mean, it's kind of like duplicating your genome and saying, I don't need polymerase now. Like, no, you kind of need polymerase. Or I don't need the sugar metabolism gene. No, you kind of need all these genes. It's just that you, and here's the trick. If it gets lost in this copy, evolution is like, oh, this one's important. I'm going to keep that copy. Okay? And if it gets kept in both, it could lose one of them. But the moment, immediately, the moment one becomes non-functional, the other copy is suddenly essential. Everybody with me? So that's kind of cool about whole genome duplication. The fact that you actually had all the genes in 2 copies, and you just didn't need them all in 2 copies. But here's what's kind of cool as well. There's some genes that were actually preserved in 2 copies, and now you can evolve new gene functions, right? And here's what's completely mind-boggling. This thing here allows you to paint this duplicate mapping between this region of chromosome 4 in Saccharomyces cerevisiae to that region of chromosome 12 in Saccharomyces cerevisiae. Namely, all of these genes that were in single copy before are now brought back together by this new species. I can figure out the ancestral gene order, which has now been separated out into 2 different chromosomes. And I can do that not just for one region of the genome, but for the entire genome. So that basically means there was a whole genome duplication that separates these two species. And I want you to picture yourself being in that room and saying, has anybody hypothesized this before? Because I was looking at this in front of my eyes. And it turns out that, yes, Ken Wolfe had actually hypothesized that there was a whole genome duplication in yeast about 100 million years ago. Why? Because he noticed that these genes in different chromosomes that were paralogs of each other, that were kind of similar to each other, that obviously had arisen from some kind of ancestral duplication event, were in fact not independent. That this duplicated gene between chromosome 4 and chromosome 12 is in fact in the same order and orientation as that duplicated gene, with a bunch of completely unique genes in the middle. So I give full credit to Ken for having discovered this, because he basically said the only explanation for this is a whole genome duplication event. And of course, he pissed off a bunch of people in the yeast community who had not seen it before. And they actually argued for years and decades against him. They basically said, no, you're wrong. There was no whole genome duplication. Or, well, there's at least some chromosome duplication. And, well, there's a dynamic equilibrium of duplicational loss and so on and so forth. Okay? So there was a long, long debate until we finally, just from sheer dumb luck, were able to sequence the species that precedes that whole genome duplication, and suddenly we could paint the whole thing. And the reason why Dujon and others were against that whole genome duplication is that you were only covering 50% of the genome in these duplicated regions. So there was no, basis for them to say that it's 100%. And the evidence remained inconclusive. Only 8% of the genes were present in 2 copies. How can you argue for whole genome duplication if only 8% of the genes are in 2 copies? But what we found was quite dramatic, that even when there was not a single duplicated gene, you could find this duplicate mapping, right? Look at this region. You basically have, 10 unique genes here and about 10 unique genes there. None of them are similar to each other. And I would never know that they're mapping to the same ancestral region unless I find the ancestor whose gene order, not gene content, but gene order, tells me about the complete duplication of that region. This example also shows you something very cool, which is that the centromeres, the places where these chromosomes come together to align and then divide, are in fact mapping in the same locations. That basically this chromosome became 2 chromosomes. Raise your hands if you think this is kind of cool. Awesome. So you can basically recognize these 2 regions solely based on the gene order, and then map the entire genome onto itself. So now I'm only showing Saccharomyces cerevisiae, baker's yeast against baker's yeast. Turns out the yeast that you use for your wine and your bread is a descendant of the whole genome duplication. So we now had 145 blocks that covered 88% of the genome. There was no more doubt that there was a whole genome duplication. So we could basically paint the series of events. There was an ancestral region, an ancestral gene order. That ancestral gene order is now preserved in Chloromasses waltii, and then that duplicated, followed by gene loss, followed by condensation of the remaining chromosomes with loss, complete loss of the intervening sequences, which now look like unique genes. And at the end, only genes 3 and 13 are kept in 2 copies. Happens to be my birthday, yay. So I made this figure for the paper. It was kind of cool. And a friend of mine sends me a copy of his textbook a few years later. It turns out that this picture is now in textbooks, which is pretty rewarding. So that was yeast. Yeast had a whole genome duplication. Most of the genes are still showing 1-to-1 mapping. 92% of the genes are unique. And in fact, when I emailed Bruce Beeren at 11 p.m., he replied at 11:05 p.m., which is quite remarkable, basically saying, how big is the genome? And of course, that got me thinking, whoa, I should have expected this genome to be half the size of the other ones, but it's not. So what's going on here? And what's going on is, again, this massive loss. So yes, you take this region, you duplicate it, but then each one keeps only half the genes and then eventually gets condensed down. Sounds good? So that's sort of the condensation. And that's why Saccharomyces cerevisiae is 12 megabases, and Chloromyces walti is only 10.8 megabases. It's not 12, but it's also not 6, okay? Because 92% of the duplicated genes were in fact lost. Yes?
Is there any kind of like what kind of genes were preserved in the duplicate?
What a great question. That's the next section. Great question. All right, so that was yeast. A year later, we collaborated with a French group to basically show, with Olivier Jaillon and others, to basically show that there was in fact a recent whole genome duplication in a vertebrate species, that this fish has in fact undergone a whole genome duplication. And for a single cell organism, you can kind of see how it can happen. But for a vertebrate to basically undergo a whole genome duplication, it's kind of weird. You're like, okay, well, who are you going to mate with if you're whole genome duplicated? And, you know, how are you going to do this? And what happens? And so on and so forth. So we could basically show that in fish, there was, in fact, another recent whole genome duplication. And then that was using exactly the same kind of signature. And this is that duplication here that now has 2 copies compared to 1. So there's a 2-to-1 mapping between human and these species. So we basically have half the gene content, more or less, or half the chromosomal content that they do, again, followed by a lot of loss. But in fact, it turns out that we have also undergone 2 rounds of whole genome duplication. And that was the 2R hypothesis from the Hox clusters. And, you know, just at the root of this vertebrate lineage, there was a whole second whole genome duplication. And just at the root of these lineages, there was another first round of whole genome duplication. So again, this is now a general signature. There's many, many species that have undergone this whole genome duplication. And you can just imagine that there are some regions of these fish that have 8 copies because, you know, all of them were in fact preserved. All right, so there was a whole genome duplication event. Okay, so we can resolve this debate. But now the question is, like you say, which genes were kept in 2 copies? So here we are. We basically go from 5,000 genes to 10,000 genes overnight. And then over time, you lose 90% of those duplicated genes. But you end up with 500 gained genes. That allows us to now study evolution and basically see how do new functions emerge. So 457 genes were kept in 2 copies, accounting for, of course, for 1,000 genes as a result of selection. And they are involved in sugar metabolism and fermentation, exactly what you would expect. Why did this whole genome duplication happen? Well, the why is hard to answer, probably because it was advantageous, but why didn't we kill it? Because that species must have been super, super sick when it was just first duplicated. And the answer is that it kind of coincides with fruit-bearing plants emerging in the history of evolution. So fruit-bearing plants basically decided that the best way to spread your seeds is not to just spread them in the wind and hope they land somewhere nice, but to make a super juicy fruit full of fructose surrounding your seed, get the mammals to eat them, and as the mammals walk around, and eventually my son will love this part, poop. Sorry, yes, I said poop in lecture. The seed is now transported somewhere else, right? That's why fruits are so delicious, because plants want us to eat them, okay? So next time you're eating a fruit, basically remember that you're just carrying the seed for that plant. So there was all this sugar in the environment, rotting everywhere, just sugar, 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 sugar. Great place for a species to say, ooh, I'm going to make 2 copies of myself. And now I'm going to have all these extra genes that I can use to metabolize this sugar. And if you go in the fields of Tuscany, in the vineyards, and you ask what lives in the grapes, it's baker's yeast. It's the descendant of this whole genome duplication. It's not some, you know, schmo that hasn't duplicated. It's really the superorganism that emerged from that now has 2 copies of all the good, useful stuff. So that answers your question of what actually, you know, was kept. But the next question is, what about neofunctionalization and subfunctionalization? So there were a lot of evolutionary theories about what happens after genome duplication, after gene duplication. What's really cool now is that we actually have an outgroup. So we can use that outgroup to root this 3-way comparison and figure out which of the 2 copies evolves faster or slower. Everybody with me here? So that's exactly what we're going to do, and look for evidence of one copy evolving faster or both copies evolving faster. And what you find is that 20% of the duplicated genes show acceleration, and in 95% of cases, only one copy evolves faster. And that basically argues for a model of subfunctionalization, where basically only one of the two copies takes the new function, whereas the other one preserves the old function. Everybody with me? So basically, in this example of this ORC1 and SIR3 are two genes, one involved in silencing, the other one involved in origin of replication, we can now infer that, in fact, the origin of replication complex is probably the ancestral version, and the silencing is probably something that evolved from it with a fourfold acceleration. Everybody with me? Similarly, this antiviral defense protein appears to have evolved from duplication of this translation initiation protein. So again, you want to sort of bind RNA and start translating, and you can co-opt that function to now bind RNA and repress a virus with SCI7. So this asymmetric divergence allowed us to recognize ancestral and derived functions. So the other question that we can ask is, are these derived genes showing different properties than these ancestral genes? And the derived genes are never lethal. If you delete these longer branch genes, the species is perfectly happy. But if you delete these slower genes, the species is actually very unhappy. So these are lethal at the rate of any other single copy gene, but these are never lethal, which is kind of cool. So you basically seem to be gaining a new function, but you know, these genes are now losing their ancestral function. And if you look at their expression patterns, the ancestral function genes are much more abundant in expression, whereas the derived function genes tend to be more specific in stress or starvation. In terms of localization, these are found everywhere, whereas the derived function are very often localized in specific subsets of the cell. Everybody with me? Cool. So we could resolve the genome ancestry and infer that indeed there was a whole genome duplication followed by massive gene loss, and we can understand the emergence of new functions by asymmetric acceleration, distinguish ancestral and derived functions, and also learn about genes that were both kept in two copies but are slow evolving. And one of the functions for that could be just simply a repository where you have a backup copy if you need it, or gene dosage and other things. Basically, not everything took on a derived function. All right, so we talked about the foundations of building the genome alignment. We talked about the foundations of, the whole genome assembly, the foundations of building the whole genome alignment. And we talked about these mechanisms of rapid genome evolution. And we also talked about these dramatic evolutionary jumps of whole genome duplication. Who feels that they've learned stuff? Yay, awesome. So now let's talk about the section of the population genetics lecture that I didn't get to cover. Yes.
Quick question on like the whole genome duplication.
Yes.
It's like one single organism like duplicated their entire genome, like one like evolutionary genome. How do they not just like immediately die?
Yeah, what a great question. So yeast has this ability to live in either the 1N or the 2N state. So we make sperm and we make eggs, and the sperms and the eggs cannot just go off having a party. Okay, they can't live on their own. They only live inside our organism. Yeast can basically become a sperm and then goes off replicating as a sperm, has a great time, reads books, etc., and then meets an egg, decides to sort of blend together and now go off, you know, I don't know, exercise, cycle, you know, play the piano, etc., and then produce gametes, basically, you know, single cell again, a sort of 1N again, and then go off living that state again. I mean, they're not male and female, obviously, they're alpha and A, so there's a mating type locus. It's kind of cool, actually. When I was aligning these species, I realized that there was one place where I was trying to calculate the rate of evolution by dividing the amino acid substitution rate with the synonymous substitution rate. And I found that my program crashed for one gene. I was like, what's going on here? It's just, I was dividing by 0. There was no changes at the nucleotide level. When I looked at what are these genes, they were the mating type locus. basically there's a single locus in the yeast genome that decides if you're male or female, if you're A or alpha. They have actually both copies. They have the A and the alpha version, and they're both silent, and they copy them in the middle, and whichever they copy, they decide to become that mating type. So if I'm A hanging out at a party and all I see is A, I'm like, well, haha, I'm alpha now. Hello. So they can actually do mating type switching. They can actually basically decide to change their gender by sort of their environment. And anyway, so they're super, super cool species. So the question is, when did the whole genome duplication event happen? When it was like reading books as 1N, or when it was cycling as 2N? And it's unclear. One possibility is that two different 1Ns that were kind of diverged from each other came together and then duplicated, or two different 2Ns that were kind of like each other came together and made it. So it's kind of unclear because this happened a long, long time ago. So Ken Wolfe has written a series of papers postulating different hypotheses. They've kind of changed their mind in review for some of these papers, and I don't know how I would know that. But anyway, so there's still ongoing debate as to when did it happen. And that's for yeast. Now, what about these fish? Again, like you said, you're kind of lonely. If you're the only person with 46 chromosomes and everybody else has 23 chromosomes, you can't quite mate. So one possibility is, I actually don't know. I don't know. Maybe the fish that basically made, I don't know, 1,000 eggs had these whole genome duplication happen before the 1,000 eggs spewed, and now you have 1,000 copies of that, I don't know, superfish. One possibility. Again, I have no idea. Everybody with me? You guys want to talk about humans for a change? Sorry, go ahead. What a great idea. So we have some examples of one chromosome duplicating in human. What is it called? Trisomy, right? So it's quite mind-boggling that you can have a whole extra chromosome and walk around, talk, laugh, cry, and all of that stuff. I mean, it's amazing that we can tolerate having an extra chromosome. Why is chromosome 21 so frequent? Because the other ones are dead. Basically, chromosome 1 trisomy just doesn't quite survive. So to be able to maintain 3 different chromosomes, I mean, a second copy of a whole chromosome, is actually very, very difficult because there's all kinds of gene dosage effects that sort of are rewiring your circuitry and so on and so forth. So this is called aneuploidy. So ploidy is basically how many copies do I have? A diploid has 2 copies, a monoploid has 1 copy, and euploid, kind of like a euphony, and all of that, means good, and then cacophony means bad. So euploidy means I have the right number of chromosomes, and euploidy means I don't have the right number of chromosomes. So aneuploidy is basically what you have when you have only 1 chromosome be there in 2 copies, and that does happen. If you look at plants, if you look at corn, they're like duplicating all over. It's amazing. So plants have like 8 rounds of whole genome duplication, a bunch of chromosomal duplications, segmental duplications. They just can't tolerate it. Chances are that it's because they don't have to just like up and leave when they're being attacked. They also can mate, like their mating rituals are just insane. Like they can have like 2 sperm and 1 egg coming together to do all kinds of crazy stuff with an egg. It's just amazing. Like the reproductive cycles of different plants. Like if you want to see weirdness, study that. But anyway, so yes, does that answer your question? Great. Any other questions? Okay, great. Want to talk about human? Awesome. You guys are so self-centered. All right. So now let's talk about recent evolution. So this is the stuff that happens in kind of like human time scale. Okay. So let's talk about sort of how are genes even emerging? How are new alleles being selected? And what did the human population coming out of Africa look like based on the recent evidence? And can we find human relatedness and use corrections for that? So this is again the second half of the population genetics lecture that you guys get because I was able to go way, way, way faster. All right, so molecular evolution, you basically have some minor allele frequency. We talked about the minor allele frequency, how, you know, this is 60%, that's 40%, that your MAF is 60%. So that's the allele frequency. But now the genotype frequency is different from that. Nobody walks around like yeast as a haploid state, right? We're all diploid. So, sorry, again, it's not monoploid, it's haploid. So basically you end up with AA or a GG or AG/GA at the frequency dictated by the allele frequency, okay? So you basically have, you know, this times that for the diploid A, diploid G, and then this is, sorry, homozygous, homozygous, heterozygous. Sounds good? So, and then of course, the question is, if A is really awesome to have, like for example, I can now drink milk into adulthood, okay? How does A rise to high frequency? Under some very strong assumptions like no mutation, no migration, no selection, large population size, allele, random mating, then A and G will just randomly drift up and down. They are, and they will eventually arrive at what we call Hardy-Weinberg equilibrium, okay? So under, again, these assumptions, a change in allele frequency of a new mutation over time is described by this Wright-Fisher model. This is basically looking at evolution moving forward. On Thursday next week, we're going to look at evolution moving backward through time. So we're going to flip around the Wright-Fisher process onto its head and talk about coalescence, namely what is the frequency that two alleles came from the same ancestral allele in the population. But now, looking at this forward process, this change of allele frequency is described by this process, Wright-Fisher. And without selection, the main force of allele frequency change is just genetic drift, so random chance. Some cases I'll go all the way to fixation, other cases I'll go all the way to fixation for the other allele, and in other cases I'll just fluctuate here for a long time, and eventually one of them will die out, okay? Why do they die out? Because this is a sync state. When I have now risen to 100% frequency, no one has the G allele anymore. So it's not like I'm gonna like drop back down, right? So basically the allele frequency gets stuck if it hits 100% or 0%. Everybody with me so far? Great. And then natural selection can change the course of this process either towards 0 or towards 1. So now we can basically ask, how can I find evidence of selection? Of sort of something something pushing me away from this Hardy-Weinberg equilibrium. Everybody with me so far? Raise your hands if you're following. Awesome, great. Okay, so now we can basically ask, where was this thing selected? So I mentioned this lactose intolerance, thing. So lactose intolerance is the norm. That's normal humans. If you can drink milk now, you're a mutant. So, and this mutation arose in Europe sometime in the last few, 10,000 years or so, 10, 15,000 years, and it was dramatically selected for. Why? Because we have cattle, because we have cows, okay? And suddenly the domestication of cattle enabled this mutation to be super beneficial because suddenly you have a new form of protein, which is much, much easier to obtain. Everybody with me here? So now the question is, how can we search for such signals of selection? Yes, we know that lactase being expressed into adulthood, the lactase processing enzyme being expressed into adulthood, was selected. First, how can we find out about that, confirm it genomically, and two, can we search for other such events throughout the genome? And there are different tests that you can carry out to measure selection at different evolutionary distances. You could look at the length of haplotypes, at population differences, at the frequency of derived alleles, at heterozygosity, at the proportion of functional changes, and these apply at different scales. Why? Because this one is very difficult to see if you're looking at very, very short distances. And this one is completely lost when you go all the way out there, okay? So we're gonna basically now describe briefly the different types of signatures of evolutionary selection. Everybody with me? So the fundamental question is, how have allele frequencies changed over time, and what are the methods to use to look at those? So one way to look at that is to basically look for deviation from this Hardy-Weinberg equilibrium of, you know, how often do you observe each of the different alleles. So basically you can test, at longer evolutionary distances, you can basically look at positive selection. So remember when I was telling you that I was looking for the ratio of amino acid changes versus nucleotide changes? Why would anyone do that? Because amino acid changes are the functional part. They basically tell you how much has the function, quote unquote, of the protein changed. Nucleotide changes tell you something about the number of mutations that have occurred, right? Because these nucleotide changes are kind of neutral in many locations, as we saw in the comparative genomics lecture on Tuesday. Actually, it feels like a long time ago, right? So basically, this bang-bang poofness comes from the fact that synonymous changes happen basically randomly. They're not selected against. But by measuring the number of synonymous changes and then dividing the number of non-synonymous changes of amino acid changes versus the number of synonymous changes, you can basically get a measure of positive selection. So this McDonald-Kreitman test basically uses that idea of looking at the number of synonymous changes and the number of non-synonymous changes, and then asking whether mutations that were beneficial to Homo sapiens were specifically occurring in the human lineage. So you can basically compare the amount of variation within the species, for example, the polymorphism, with the amount of variation across species. And you can do that for the synonymous changes, you can do that for the non-synonymous changes. And then if you find that the ratio of non-synonymous to synonymous within a species versus the ones that are actually polymorphic are in fact, you know, if there's more fixed changes that change the amino acid than polymorphic changes, that basically implies purifying selection. That basically means that you're keeping the ancestral form. If instead the number of fixed changes that are derived from the ancestor that are different, that are happening in human, are in fact greater, that implies positive selection. So that basically says that if a site that is different from what the chimp had is now fixed in the human population, that basically means that it rose in frequency and it basically became the dominant form because of selection. Is everybody with me? Great. So that's one test. That's basically looking at the proportion of functional changes, this amino acid versus synonymous site, and then the ratio of that to between the fixed and the polymorphic sites. You can also look at the frequency of derived alleles. You can basically say, great, there's a derived allele that separates us from chimp, or a derived allele that separates us, say, in Asia from the sort of African mutations that we had. If that derived allele rises in frequency, that's again evidence of selection. Everybody with me here? So you can basically measure what is the frequency of these derived alleles, look at the ancestral state, and then ask, you know, how frequent is that allele in the population, and then from that infer this. The other thing you can do is basically look at differences between populations. If a different allele rises to fixation in Africa versus Asia versus Europe, that's again evidence of environment-specific adaptations. So you can basically measure the difference in allele frequency using this FST statistic, which estimates the reduction in heterozygosity of how different alleles are from each other between two different populations. You can basically say, well, if these two are isolated populations, how often would I expect to see them mixed? And then FST basically tells you what is the overall reduction in heterozygosity by subgrouping these populations. Basically, if I think of this as being a single population, and then I group it into Asia versus Europe, for example, then the population differentiation test basically tells me how much more segregated are they than what I would have expected by chance? Namely, what is the reduction in diversity? But diversity within alleles is heterozygosity. So what is the reduction in heterozygosity by grouping the populations versus the overall heterozygosity? Raise your hands if you're with me on this one. Awesome. So basically what we're doing is asking, by separating them, how much more distinct do they get? And then the last one is actually building on what we were talking about in the first population genetics lecture about the length of haplotypes. So as I mentioned earlier, these haplotypes are simply places where recombination has not yet occurred. Okay, so you end up with very big haplotype blocks in between these recombination hotspots where PRDM9 comes and binds its beloved motif. Okay, between them, recombination is very rare, but it does happen every now and then, very rarely. And the question is, how long is the homogeneous, co-inherited haplotype? If it's very long, it's probably very recent, because over time, recombination events will tend to break these haplotypes. Raise your hands if you're with me on that one. So basically, the length of haplotypes over time tends to decay. Okay? Now you can ask, how old is an allele based on what haplotype is it sitting on, based on the age of that haplotype? And if you find a long haplotype with high frequency, it basically means that this was selected for. That basically whatever allele I have here has now been selected for. So basically the evidence of recent positive selection is when you have a long haplotype, which basically tells me that something is recent, but it has risen to high frequency. And things don't rise to high frequency very, very fast with just drift. So there must be something pushing it to high frequency. Sounds good? So I can look at the frequency of different alleles, the frequency of different haplotypes, and then how long they are. And discrepancies between having this very long haplotype and having a high frequency suggest evidence for selection. So for example, if you look at this gene that allows Europeans to metabolize lactose into adulthood, you see this dramatic long, super long haplotype at very high frequency. So that basically says that this region was very, very heavily selected for. So it encodes the lactase enzyme, and it's required to digest milk into adulthood. And there's evidence of positive selection in Europeans. The persistent haplotype is present in 77% of Europeans. So that basically means there must have been something good in that long haplotype that was very, very heavily selected for. Okay, so basically any haplotypes that don't have that particular mutation are sort of weeded out from the population. Is everybody with me? Awesome. So you can basically build all these selection tests, combine them, and then search for evidence of selection. And when Pardisa Betty and her group did that, they found more than 300 different regions that were selected, including lactose tolerance, resistance to malaria, and many, many others that they're sorting out biologically. Okay? Everybody with me? So I guess this is where we're going to end today. So we can measure selection in the human population for recent positive selection, but we can also measure selection for purifying selection. So in fact, if you look across the allele frequencies for different classes of elements, you find this change in allele frequency, which is indicative of selection, specifically for gene regulatory elements, etc. As for the, I have to tell you one more thing. You guys want to hear one more thing? All right. If I take one person from the planet, if I abduct that one person from the planet, and I take him on an alien spaceship, there's enough information inside the genome of that one person to basically tell me about human demographic history over the entire ancestry of that person. Mind-boggling, right? Why and how? Because every location of the genome has a different history. By comparing paternal and maternal, which are presumably unrelated, unless they're marrying their cousin or something, paternal and maternal are basically two different samples of the human population. if I scan across the genome for how many differences do these two chromosomes have, I can basically get an age for when they diverged. And I can age every single section of the human genome, giving me a distribution over time for how many, what fraction of my segments were diverged at what fraction in time, at what point in time. And therefore, I can infer from a single person that there were, you know, many segments coalesced there, and then many fewer segments coalesced there, and so on and so forth. So basically, if you look now at you know, what happened, you basically see this bottleneck. You basically see this expansion in Africa, this bottleneck as the out of Africa event is happening, and this recent re-expansion in the different populations, right? And you can pick an individual from each of these countries, and from one person, you can build all that by comparing across the genome the pairwise similarity. Who thinks this is kind of cool? Awesome, awesome. So who feels that they've learned stuff today? Yay, good. So we'll probably figure out some way to put this back in the lectures. See you guys on Tuesday. Monday's a holiday.